Hello, everyone. Good morning and welcome. I am Sheryl White, Coordinator of Visitor Engagement and Exhibitions here at the Arnold Arboretum in Boston. The 281-acre Arboretum landscape has collections ranging from A to Z, or in plant parlance, Acer to Zelkova, including over 15,000 plants of over 2,000 different species. Many of those plants get their start as seeds in other parts of our world and are brought here through our plant collecting expeditions. Today's speaker, Sean Halloran, is not only one of the collectors with many other staff at the Arboretum, but our propagator as well, ensuring that those seeds succeed, become plants, and continue to maintain their presence in the Arboretum's collection and in our world. Sean Halloran is the plant propagator at the Arnold Arboretum of Harvard University in Boston. He received a bachelor's in horticulture and a master's degree in plant and environmental science from Clemson University, where he focused on nutrient media in tissue culture propagation. His professional background includes private residential horticulture, greenhouse and nursery production, plant nutrient management in soilless media, and temperate woody plant propagation and conservation. As the Arnold Arboretum's plant propagator, Sean continues a 150-year tradition of growing woody temperate species from seed, cuttings, grafts, layers, and more. After Sean's presentation, there will be time for questions, so please put yours in the Q&A. I want to thank all of you for being here, and I want to especially thank Sean and welcome him as he gives us our presentation today, Wild Collection and Propagation of Rare and Endangered Plants. Sean?
Thanks so much, Sheryl, and thanks to Amy as well, both of you for coordinating this and doing all the planning. And thanks everybody here for attending. Seems like we have pretty good attendance today. So, one of the most exciting parts of what we do here at the Arboretum for, I think, many people, including, those of us who get to go on these trips, are what you're seeing here, the plant collection trips, where we collaborate with botanists, other plant scientists, and then groups that are interested in land, animal, and plant conservation, but also those local guides that really just know the areas in which we are looking for our target species. So we'll talk a lot more about this as we go along. So very simple statistic here for you, and that is that 97% of climate scientists agree that climate change is real. It's caused by humans, and the last bit of our little tagline for today is "It's not too late," and that's really what we're focusing on. I'm not a climate change scientist or subject matter expert there, so we're not going to go into the science. But if you're really interested, there's a great 2016 paper, Cook et al., that focuses on the consensus, and that's where the statistic comes from among climate scientists and science more broadly. And then you can find a lot more information about this consensus at climate.nasa.gov. And there's a lot of great papers and resources that discuss this. Also, there's a lot of great people in the Boston area focused on science literacy, and that's why we're talking about this today, so that you can learn how the Arnold Arboretum plays a part in saving plants, but also more broadly the people that we work with, the great folks that are also on the front lines of this. I want to stress the point that we can't change anything or expect people to change by just supplying them with fear and sort of this fear-ridden rhetoric. And that's why the last part is it's not too late. Those are sort of the three stages of climate denial. It's not real, it's not caused by humans, and it's too late. So I just am here to say it is not too late. Of course, there are scary effects of climate change. But again, we're talking about conservation of plants, and it's going to be a lot of pretty photos of plants and seeds and flowers here. So as mentioned in sort of the description for this talk. Plant conservation hits home to many of us probably on this call that are here in Massachusetts, but it affects us all around the world. In Massachusetts, vascular plants make up 153 of the 217, so about 70% of the official listed species that are considered endangered, threatened, and special concern. So here you can just see two plants that were with these nice fact sheets done by the Massachusetts Fisheries and Wildlife Division, just highlighting these two endangered plants. On the left, New England blazing star, and on the right, yellow lady slipper. So some recent estimates say that around 1 in 5 plants are estimated to be threatened with extinction, and some recent work puts that number maybe even closer to 40%. And again, that's the end of the fear and the sobering statistics today. But I hope that conveys the gravity of the situation. I posit that one of the hardest things in communicating about plant conservation is relaying the value that plants provide. There's a term which I'm not going to repeat today, but it's sort of an accepted term for a cognitive bias that humans have around sort of plant ignorance or not appreciating plants. I have termed it plant underappreciation, but because I think this is more apt. It's essentially an informal idea that there's a general tendency for humans to not recognize the importance that plants have in our daily lives, but more importantly, in complex global ecological systems. And again, that's why we're talking right now. In curating, growing, and maintaining these plants at the Arnold Arboretum and at our partner institutions around the world, we're providing a physical space where people can come and realize and study the many benefits that plants provide to humans directly, but also just the importance of saving plants more broadly. So what is a rare, threatened, and endangered plant? Obviously, there's legislation, local, state, or provincial, and federal in many parts of the world that protect animals and plants. In just like for us, the 1973 Endangered Species Act in the U.S. But in our jurisdiction, we're also subject to local governments and the state's own Massachusetts Endangered Species Act. Suffice it to say, there's as with any legislation, terminology is often different, and because of overlapping regulations, it can get very confusing. So for our purposes today, I'll just use the words rare and endangered, but you might also see special terms like special concern. So a great global resource that you might see cited very often is called the IUCN Red List, which can be found at iucnredlist.org. And in sort of the bottom left of this image, you can see the whole spectrum from left to right here: not evaluated, data deficient, least concern, near threatened, vulnerable, endangered, critically endangered, extinct in the wild, and then extinct. So obviously these are established categories that IUCN uses to categorize plants. And you can see here the little oak seedling that I showed earlier growing at our greenhouses right now. You can see that Quercus acerifolia, or maple-leaved oak, is considered endangered. And the red list basically goes on to provide published scientific literature as to the breakdown of the threats to this particular species or taxon as being land use change and degradation, potential destruction of habitat, and also scientific specimen collection. It goes on to say, quote, that a subpopulation which holds more than half the total number of individuals lies on privately owned land where no protective status or conservation agreement exists, end quote. Furthermore, they talk about secondary threats, suppression of fire. This is something that we encounter quite a bit, has led to a decline in the early successional open canopy woodland in which Quercus acerifolia thrives. So with that said, let's just talk a little bit about this term ex situ plant conservation, which describes what we do here at the Arboretum. Ex situ essentially means off-site. So in very basic terms, ex situ conservation is the process of protecting a plant species or a variety of plant outside of its native habitat. Obviously, the best thing is to protect the habitat, right? And have no, human interference and so on and so forth. But obviously, in a lot of areas in our country and around the world, this is not possible. So what we do at the Arnold, because we're an arboretum, is sort of a conventional form of ex situ conservation. You might also imagine other botanical gardens that we work closely with, or even in the case of animals, you might imagine a zoo is a place where ex situ conservation takes place for animals. So in the case of the oak that we discussed, we would, responsibly collect acorns from those oaks so that we didn't damage the existing populations going forward, and then grow them on here at the Arboretum. And we'll talk more about that later. Obviously, we're going to focus on propagation. So not only do these institutions function in informing the public about the factors that might lead to habitat loss or the tenuous position in which a species might be in, but obviously we also keep that genetic material alive, off-site, ex situ. So when you walk around the Arboretum, keep in mind that, every plant has a story, and many of those stories involve a conservation component. And we'll talk about some recent work by Catherine and Michael in our curation department, looking at the assessing conservation status for a lot of our plants here. So this is a photo of the Millennium Seed Bank at Royal Botanic Gardens, Kew, where they store about 2.4 billion seeds from 90 countries. And you might be more familiar with this idea of seed banking. And so they and other seed banks prioritize storage of seeds that can tolerate dry down and long-term storage, something that we generally refer to as orthodox seed. You're probably also familiar with the global seed vault in Norway called Svalbard, pictured here. Again, many seed banks prioritize plants, especially from areas that are very vulnerable to habitat loss due to human encroachment, as well as wild types of agricultural crops, corn obviously being one that comes to front of mind, and plants that are rare, threatened, or endangered in the wild. I want to make clear that there's no one way to do ex situ conservation. Seed banking is important. Obviously, what we do here at the Arboretum is important, but it takes a lot of institutions working in different ways, using different techniques to create to conserve these plants. And we'll talk about some local resources at the end, some folks that are working towards this. So as I mentioned, it takes efforts of many groups of people working together to save these plants. And one way that botanic gardens and arboreta especially collaborate with one another is via these coordinated plant collection trips. So I'm going to use our most recent trip before, obviously, the global pandemic as an example here, because this was truly an international affair. Keep in mind that many of these such trips and there are really too many groups here for me to name. I'm just going to give you a few. But there are many such organizations that promote these kind of collection trips. So if you've been following the Arboretum for a long time, you probably know that folks from the Arnold have been collecting in China for the past 100+ years. And most recently, our keeper, Michael Dosman, and our head of horticulture, Andrew Kopinski, have kept this tradition alive and well. While in 2019, roles were reversed when Jared Rubenstein, then fellow, now project manager, Andrew, and myself were given the opportunity to plan a 3-week expedition in the American Southeast for 3 of our longtime Chinese collaborators, Doctors Kong Wang, Xinfen Gao, and Tao Dang. And the whole the whole group is pictured here. So we all spent months preparing for this 3-week-long journey, which began with identifying target taxa, or those species that we were looking for. We identified taxa that our Chinese collaborators were interested in, both the Beijing Botanic Garden and the Chinese Academy of Sciences. And we selected plants that were able to grow in their respective gardens in China, or that they were able to store via herbarium vouchers or dried plant specimen in their herbaria. Angela Magnan of the U.S. National Arboretum and her garden also collaborate on this trip. And the National Arboretum is in Northeast Washington, D.C., and is administered by the USDA ARS, the Department of Agriculture Research Services. So now with all these institutions on board with different missions and interests, we had a big plant list, and we're all aligned in the name of plant conservation. We set out to define a broad geographic area over which we would collect. And that ended up being very broad, being Ohio, Kentucky, Tennessee, Georgia, South Carolina, and North Carolina. So a huge swath of our country that sort of covers the border of the Ozarks, and also a large swath of Southern Appalachia. And this is where we can find some of the highest plant biodiversity outside of the tropics. So it's an incredible region to collect in. So obviously, one amazing tool I mentioned earlier, Herbarium Vouchers, for finding the locations of specific plants that have been collected historically, is Herbarium Vouchers. And this gives you a ton of information about, in this case, a 1942 collection of Acer rubrum in Havelock, Ontario, Canada. And we traditionally have used these kind of records to plan these trips, right? We know that there's an existing population of this red maple that we're looking for, and we're focused on, let's just pretend, this part of Canada, and we want to find a subpopulation of this plant, so we know exactly where to go. There's a description right there. But obviously, things evolve, and we have very powerful tools like GBIF, or the Global Biodiversity Information Facility. They provide free and open access to biodiversity data like this that greatly aids in conservation efforts, and especially in mapping out these confirmed science grade observations. So again, you can find this at gbif.org. So with the advent of a tool called, and there's also other tools, but iNaturalist is one that you may have heard of, basically allows citizen scientists to make observations about plants that they see. And then tools like GBIF that I mentioned are able to collaborate, grab all of that data, including historical herbarium vouchers, and put it all together. And so it's extremely powerful when you have, citizen scientists working together, and then tools like iNaturalist, which can really, provide a scientific level observation that is vetted by several people. So now that we've sort of identified, okay, these are the hotspots we want to focus on, we had to get to work then identifying specific public and private lands where we could find these plants that we're interested in. I just show a map here of basically all those points that we collected from GBIF, and then overlaid them on obviously a current map for our target taxa. And you can see, this massive swath of land that we were focusing on. And so we use this to hone in on specific places like Daniel Boone National Forest, where we would make connections and contact folks. So again, it's all about making these important connections with land conservation groups on the left, Beverly James and Josie Miller were our contacts at a nature preserve in Kentucky. On the right, Emily Ellingson and Rob Peratley are pictured here with our group. And they were, vital in our collections in Daniel Boone National Forest. So we had people who were very knowledgeable to guide us, but we also had to obtain permitting. In this case, with the national forest, have to obtain permitting through our national government. And it's incredibly important to make those connections with folks, as well as, other local, state, and federal agencies, so that we're in compliance with all the laws that are in place. We had so many collaborators on this trip. Obviously, I couldn't name them all here, but really, these are the people who are working on the front lines of setting aside land, first of all, and then getting the word out about saving plants and animals in their respective regions. So here, this is just a very brief over, a cap of what we did, our plans and where we traveled. Like I said, we collected in Southern Ohio, all throughout Kentucky, down into Tennessee, up into, again, the beginning of Southern Appalachia here. And then we made our way back home to Boston with these 100 different accessions that we collected over this 3-week period. And, now it was time for the propagation work to begin. So as we were going along, we ended up cleaning seeds, shipping seeds back to the Dana greenhouses here where I am today, where our staff, properly labeled everything, cleaned things, and the seed, inspected seed for any insects and so on, assigned the proper treatments, which we'll talk a little bit about in a second. And then they were quickly whisked off to be grown here at the Arboretum or sent to our partner institutions like the U.S. National Arboretum, Beijing Botanic Garden, Chinese Academy of Sciences, and many more. And so I just mentioned seed treatments. What I mean by this is basically the set of chemical, environmental, and even mechanical means by which we germinate these seeds. In my seed lecture, I'll often say that a seed is a baby in a box with lunch. That's just sort of how the very basic model I present you to understand how seeds work. So the baby is the embryo, you can see in green here. The box is the seed coat, and the lunch is basically the starchy or fatty component, often what animals like humans are trying to eat when we eat seeds. And this is important to understand in the context of plant conservation, because those aforementioned treatments are reliant upon understanding the basic seed biology and the specific requirements that the embryo, the baby, the seed coat or the box, and the lunch or the endosperm all have for the species that you're attempting to germinate. So, for example, some oak species, like pictured on the right here, produce acorns that are not considered orthodox or traditional seeds, because as soon as they start to dry down, they also begin to lose viability. So many oak seeds are considered recalcitrant and have to be stored at a higher moisture level than many orthodox vegetable seeds, for example, which can often be stored for years at very low moisture levels or very dry. So all of this is to say that we do a lot of research based on the scientific literature. So people publishing germination data and treatments and so on, our own robust internal propagation records. And then obviously, as I've really tried to hammer home, conferring with colleagues and other folks who are working in plant conservation or plant propagation to help us make decisions about how to germinate a specific taxa. So for limited quantities of rare and endangered plants, the pressure is really on for us to make the conditions just right for successful germination. And of course, imagine that we have multiple trips per year, maybe a trip to the American Southeast, a trip to Japan, a trip to China, and then perhaps hundreds of these decisions to make every year. Really, it's a fun logistical challenge that I really enjoy. So our germination treatments were successful, and now we have viable seedlings that we can transplant from our greenhouses out into our in-ground nurseries. And then after 3 to 5 years, these plants move on to become members of our living collections. So our incredible group of horticulturalists plant these trees and shrubs out in the landscape in the spring or in the fall. And then, they work year round to protect the health and the well-being of these plants as they live out their lives. But a big part of ex-situ conservation is preservation of genetic material. And since most plants live only decades or perhaps a few centuries if they're lucky, we must inevitably propagate them. And because in most cases we want to preserve the genetic material that we encountered in the wild, we generally perform what we call asexual propagation on the plants in the living collection. So rather than collecting seed, as we would from wild populations, we're taking cuttings, grafts, and in some cases using layering to save these historic plant collections. So these techniques can also be used in ex-situ conservation from the wild, especially in sort of extreme examples where plants are threatened in a very time-sensitive way. So on the left, you see that you see a rooted cutting. Basically, cuttings are made from vegetative portions of the plant, such as stems, modified stems like rhizomes, tubers, corms, bulbs, and then leaves or roots. Those are all cutting types. So cuttings are generally classified by the part of the plant that they were obtained from, or sometimes the developmental or seasonal period during which they were harvested. So, for example, you might hear the term stem cuttings or hardwood cuttings, or in the case of the plant on the left, narrow-leaved evergreen cuttings, and so on and so on. The type of cuttings and the treatments that we assign to those cuttings will largely depend upon the species that we're attempting to propagate, but also the time of year or the season. And generally the main goal here is development of adventitious roots that you see here in the leftmost image, or roots that arise from the aerial plant parts. In the case of root cuttings and underground structures, generally you're trying to develop shoots and roots from those underground stems or root pieces. So in the center here, you can see a graft. Grafting is a traditional art that humans have been practicing for about 3,500 years. And it brings together 2 or more pieces of living plant material to serve some human need, be it ornamental, cultural, agricultural, or in this case, for plant conservation, clonal. So most grafts consist of a top part, the scion, a graft union where these 2 genetically different plants meet, and then the rootstock or the desired root system. In the case of rare and endangered plants, sometimes grafting is really the only viable option that we have, just depending on how easy to root this plant in question is, or perhaps even time constraints. We do have storms here, obviously, and plants do come down. And so there might be a rush to, let's just try to keep this material alive. And sometimes grafting is the right thing to do there. So the image on the right shows our third type of asexual propagation we're going to talk about today, broadly called layering. And what is pictured here is specifically called air layering because it's done up in the air rather than down on the ground, like more quote unquote traditional techniques of layering. So air layering is very simple, but highly effective technique for essentially rooting cuttings in place while still being attached to the parent plant. So we've been working on a research project for the past 4 years in the landscape, basically trying to figure out which plant genera and specific taxa we can air layer successfully. And this is a really exciting, part of the propagation work that we do. And it's already led us to obtaining clonal material of some rare and threatened species. So if you're interested in learning more about any of what I just talked about, we do teach a number of propagation classes throughout the year. Check back with our calendar as we put things there. So I just want to wrap up by talking a bit about local resources in Massachusetts. I'm assuming most folks are here. We did mention some global resources, but one resource really close to home is the Native Plant Trust, Garden in the Woods, and Nasami Farm, where they do a lot of the propagation. This is the organization formerly known as New England Wildflower. And, they have some great resources like this page I'm showing you. It says, what can you do? Another resource is Grow Native Massachusetts, which is grownativemass.org. It's a great organization that focuses on protecting our native plants and encouraging folks to plant them in their yards. Of course, Mass Audubon at massaudubon.org. Here's a great fact sheet about native plants in Massachusetts, and then the animals that depend upon those native plants. They also have a focus on threatened and rare animals and plants as well. And of course, our state has a lot of great resources, mass.gov. They obviously maintain the list that I referred to in the beginning of rare, threatened, and endangered plants. And they also have, a few fact sheets like what I'm showing here, native shrubs for planting and wildlife food. And of course, if you've ever heard me give a talk, I'm going to plug the land grant. UMass Amherst has some really great land conservation tools for folks who are trying to get grants or trying to set aside land. There's some really great tools here at ag.umass.edu. Go to their resources page and you can find a lot of great information there. And of course, I could not get away with not plugging something great that was just released, I think, last week or a few weeks ago. I mentioned before, Catherine Richardson and Michael Dosman released our conservation review of the collections, and basically found that around 10% or 1,200 individual plants of our living collections are threatened in the wild. Catherine and Michael highlighted in particular Quercus acerifolia, which is, we talked about that a bit today. They also highlighted white ash or Fraxinus americana and the devastation caused by the emerald ash borer, which many of you are probably aware of. But a staggering fact from that great article they wrote is that within about a decade, white ash went from being globally secure to threatened with extinction because of this pest. The Arboretum is working to preserve living germplasm collected in the wild of green ash, and also herbarium vouchers from our living trees. They also focused on Buckleya or pirate bush, which is one of the collections we made in 2019 on the NACPEC trip I mentioned, as well as Australia raderiana. It's a hop hornbeam from China, which we're trying multiple asexual propagation measures like air layering, cuttings, grafting, and our fellow Christine Ventura is working on etiolation of that taxon as well. So lastly, chances are wherever you are in the world, there are probably some great people fighting for land, the plants, or the animals. Go out and find them. And with that, we'll take some questions. Seems like there's a few already in the chat. And, thanks for coming today and listening to me drone on about something that I love sharing. So thank you.
Great. Thank you so much, Sean. That was really wonderful, informative, and I just think great for all our many, many listeners. So we have a couple questions. I'm going to try to do my best with one of them, from Elaine or Eliane, perhaps. If you speak of genetic conservation, do you genotype or sequence plants in order to monitor issues like bottleneck effects in breeding, when not many plants are part of a reproduction cycle, since they are rare, and or have an idea how big the gene pool is for these rare plants?
Yeah, this is not my area of expertise, but certainly there are some great people here at the Arboretum who are working on a similar work to what you describe, and then there's a global community of folks who are focused on this kind of thing. But everything you described is going on, and it's generally a familial or a generic by genera sort of research, but yeah, it's a very good question.
Great. And I actually want to introduce Amy, who is working on the panel with us. She is our digital content and program manager. And so we will be back and forth with some questions.
Mm-hmm. Yeah, hi everybody. So the next question comes from Therese, and she asked, would it be possible to have a copy of that plant list? If that's something that we can share, Sean, we can put it in that follow-up email that we'll send.
Sure. Yeah. We have all of our expeditions. I'm assuming you mean the plant list the plants that we collected in 2019. All of our, we'll give you a link to the expeditions unveiled, where you can read the blog post that Jared wrote. You can see that list of all the collections we made, and in some cases you'll be able to see where those plants are now. So that's a great question.
Great. And a nice, awesome job, Sean, as we all believe. So when is your next trip? As we open up, are there talks about going back on the collecting trail?
That's a great question. I think there are talks, but nothing as yet to report to everybody here today. I can't wait just, to share that with you.
Yeah, I actually, because I know that the campaign for the Living Collections is a 10-year project, and I wonder, and I'm, just asking you as one of the people who do collect, if having this year of the pandemic and the guidelines that we all had to follow, if that year will actually just be a regular year, or they'll add a year onto the 10-year. Do you know?
I don't know specifically about that, but I know that, some of the strategic planning that we did was to front load the collections, because of course things happen. No one in the world expected this to happen, so perhaps we'll give ourselves some leniency there, but we were ahead of schedule in years prior, and so I think we're in good shape. But yeah, we'll see what happens there. Sheryl, good question.
Great. Amy, I'll just turn it over to you.
Yeah. So next question, who has the best list of endangered species to plant in small city gardens, if people are looking for a resource around that?
That's a good question. So, like I mentioned, Native Plant Trust, they grow a lot of plants. I would say check out their resources, check out Mass Audubon, basically those few things that I showed at the end. And again, we'll put a list of those links together for you. And if you can't find that information at those sites, let us know and we'll point you in the right direction.
Thanks. And Marie has asked, what are the worst invasive plants that we should watch out for? That's a tough one.
Yeah, that is a tough question. I worked a long time in landscapes in Metro West Boston, and so just speaking for this area, obviously Japanese knotweed is a huge issue for us. I'd say that's in a lot of areas that's number one. our list, but it really depends on where you are in terms of invasives. You know, the state of Massachusetts also has a prohibited plants list, so there you can find lists of especially invasive plants. But again, it really depends on where you are specifically. Tons of great resources out there.
Right, right. For each state, you know, go to your own state's resources because they are different state to state.
Absolutely. And again, shout out to land grant universities in your state, you know, URI, UConn, UMass, UNH, UMaine, wherever you are.
Michigan State.
There you go. Yeah.
Cornell. Mamie?
Exactly.
Okay, so another question comes from Gary asking, do you think that the increasing amount of policies such as the Kyoto Protocol are hindering the speed in which plants can be conserved? Noting that he says this in regards to how quickly ecosystems are being degraded in places such as California and Australia due to fire.
Yeah, again, I'm not a climate scientist. I don't focus on that aspect of things. So I'm not going to speculate on that really. I think I'll just speak from my own personal experience. I don't think regulation is necessarily the issue in a lot of cases, but it's a good theory, Gary.
Great. Well, we don't have any other questions, but I want to thank everyone so much for coming. I particularly want to thank Sean, who is doing great work for the Arboretum, and it's actually great work for the planet. So I want to thank everybody. Have a wonderful afternoon, and check out our website for other events that we have. Thank you very much.
Thanks, everyone.